Okay, welcome, ladies and gentlemen, to Natasha's inaugural. I'm Stephen Hales. I'm acting as Head of Department of Computer Science at the moment in John Shaw-Taylor's absence. This is one of the nice bits of the job. Okay, so to be able to introduce Natasha, sit and listen to her. She does amazing research. She's a great ambassador for computer science and a whole range of other disciplines as well. So welcome to everyone here. Welcome to friends, family, supporters, colleagues. It's good to see everybody here. It's good to see a good turnout. A few words about Natasha. It's an opportunity to celebrate Natasha's achievements. It's an opportunity to hear from Natasha. Those of us who've not heard from her before, it's an opportunity to hear from her about what her research is and to learn something from that. But Natasha's an interdisciplinary researcher. Within the department, we've got really quite a strong ethos of interdisciplinary research. But Natasha personifies that. She works in areas from computer science to mathematics to biology to medicine and to economics now. She likes a challenge. Fundamentally, her research is about systems. It's about interconnectedness of things. It's about the connections between things. It's about understanding how biological systems work as a whole. And that's one of the big challenges of today. Natasha's been recognized very widely as an expert in this area. She's achieved plaudits from a recipient of the Needham Award. She's got an ERC grant. She had an NSF grant corresponding to the ERC grant. And she's one of the young Academy of Europe as well. So, I'm not going to say terribly much more. I'm going to invite Natasha to the stage, and we'll hear from Natasha on data-driven medicine. So, thank you, Natasha.
Thank you. Thank you very much for your kind words, and I'd like to thank the department for organizing this wonderful event. Today I will present some of the work that we've done in my group over the past 10 years or so. This is an overview of what I'll talk about. As we have heard, I view biology and medicine as a complex world of interconnected entities that we need to mine to extract biologically and medically relevant information. So first I will give a brief introduction, talk about the data and the main challenges, why we are doing what we are doing. Then I'll give some examples of the new methods to mine the interconnected world of biology and medicine. I will talk mostly about molecular networks in the cell, but also I'll give an interesting example from economics, a couple of our recent papers on the analysis and tracking the dynamics of the World Trade Network. Then I will show how, given the analysis of each individual layer of the data, how we can fuse them, how we can integrate them to learn about things that we care about, to personalize medicine, to classify diseases, to do gene ontology, meaning to assign annotations to unannotated genes and so forth. And if there's time, I'll point to some future directions. So let's start. We've been increasingly amassing large amounts of data in all areas of human endeavor, and biology and medicine is no different. So basically, we have collected large amounts of genomic data, epigenomic data, transcriptomic, proteomic, metabolomic, all sorts of omic, phenomics, exposomics, metagenomics, which is basically bacteria in your gut, and so forth. Huge amounts of data are available that are deposited in many of the publicly available databases, biobanks, and so forth. But each of these types of data gives basically one-sided view into the system that we are studying, which is the cell, the tissues and the organism in general. So for example, transcriptional regulation describes which proteins influence expression of genes that make some other proteins. Then we have protein structure networks, basically what is the three-dimensional structure of these biomolecules that are vital to life and function. Then we have protein-protein interaction, basically physical bindings of proteins, because proteins don't do things in isolation in the cell, but through bindings with other proteins, they change their three-dimensional conformation to do a particular task. Then we have all sorts of genetic epistatic interactions where single mutants give a different effect than the double mutant, basically mutations of two genes. Then we have networks describing all metabolic processes in the cell, and so forth, and so forth. We also have ontologies describing, for example, diseases, how diseases are related in human, and if you have a particular disease, whether you are more likely or less likely to have another disease. We have various phenotypes in many of the publicly available databases. For example, the UK Biobank, you can apply and download about 500,000 of patients at the moment, full electronic records, including genomic and proteomic data. Then there is GTEx database, where basically you have, well, relatively healthy individuals who are dead now, but gave their organs for transplantation, and basically over 47 of their tissues, you have full omics layers of all of these types that you can download, and I believe this is around 1,000 individuals right now in this database that you can download. So all of these basically give different views into the system. Each one layer in isolation is myopic and biased, but with all of them, we need to look at our system and study to actually figure out the collective behavior and what this actually all means. Now, we design tools to mine these wealth of data, these large data sets. Now, why do we need tools to mine them? So for a long time now, we've had genetic sequences, and we've built many tools, and they've revolutionized our understanding of biology, of disease, of evolution, and so forth. But while dealing with genetic sequences is computationally easy, meaning that the answers we can obtain on these sequences in a small amount of time, polynomial time, dealing with large networks is much more complicated. And most of the problems we are asking to answer from these data fall into the so-called category of computationally intractable, NP-hard, NP-complete problems. And one trivial example is comparing two networks. Okay? Stephen Cook in 1971 has proven that subgraph isomorphism problem that tests whether one network exists as a copy in another network, that's NP-complete, meaning we cannot solve this exactly on large data, even given all the time of the universe and all the compute power we can possibly get. And this is why we need to design approximate ways, algorithms to approximately solve these things, because, yeah, we want to compare a protein interaction network of a healthy cell and diseased cell to see what went wrong. We want to compare these networks over different species to infer evolution and so forth. But an inherent property of every approximate solution is that it is guaranteed to fail on certain examples. And this is why we need to build sophisticated methods that are carefully tuned to extract domain-relevant knowledge. So basically that they work well on a particular domain, because for sure they will fail in certain situations. And this is why people are talking now about these embedded data scientists and not like generic data scientists, because you cannot really take somebody who was doing statistics on fisheries in the rivers of British Columbia and bring him in a medical informatics lab and expect the person to perform well. So all of this is very domain specific. All right, so we build methods to extract information out of each layer of interconnected data in isolation, to learn as much as we can from each of them individually, and then also to fuse all the data together to see what they collectively tell us, and furthermore, to do it in a patient-centric way and to figure out how to modify treatments for a particular patient, how to customize treatments for particular patients. All right, so now, after this brief introduction, let me give some examples of the methods for analyzing a single layer of data. And I'll focus mostly on molecular networks. Okay, so the cell is one of the most complex molecular machines that we've encountered so far. Each of basically these layers of information interact between each other, whether it's genes, whether it's proteins, metabolites, lipids, and so forth. So they interact within one layer, but also across the layers. Now I'll talk about just one layer, just one network, let's say protein-protein interaction network. Now, in this network, we model proteins as nodes. This is basically a way to abstract information for easier computation. So the nodes are proteins, and if physical binding between proteins is possible, then we put an edge. So this doesn't mean that this network will exist in every cell. No, it won't. This is just what's possible. This is possibilities. And then presented with another network, we want to be able to compare them, as I said, between healthy and diseased cell, let's say. But we cannot do that exactly. So we resort to some simple heuristics, such as we compare the numbers of nodes of those networks, the number of these edges or links between them. We see how many links each node has, and we plot the distribution of those links over all nodes, so this is so-called degree distribution. But actually, these are very simple approximations of what we are trying to look at. And this is a simple example. It's very easy to construct for each of these heuristics the generate example on which your method will fail. For example, if you have this network G and a network H, they have the same number of nodes and edges. And each node in each network is of degree 2. It has 2 neighbors. So that their degree distribution is identical. So these measures will tell you that these are the same, but you can just eyeball them and see how different they are. One is connected, one is disconnected. So you cannot use these to do any meaningful analysis, really. And this is why, quite a long time ago now, 13 years ago, I introduced so-called graphlets, small subgraphs of networks, as building blocks, kind of as Legos of these large networks. And I will not go through the details. Basically, they're induced subgraphs, meaning that once you pick the set of nodes in the network, you cannot pick just a subset of edges. Because if you picked these 2 edges, you see one 3-node path, you see the second 3-node path, the third 3-node path. While if you pick all the edges, you're dealing with a triangle. And whenever we are trying, I mean, my background earlier on is my past life. I was a mathematician. I did graph theory. I proved theorems on the structured families of graphs. And whenever we are trying to understand the structure of any network or a graph, we deal with induced rather than the partial subgraphs. Okay, but that's perhaps too technical. Now, what I'm trying to do is I'm trying to figure out how these building blocks, these graphlets, these Legos, are put together in the data, and whether there is something we can figure out from the way they're put together. So basically, once we know how they're put together, whether in the end we can figure out whether we have a pumpkin or whether we have a helicopter over there. And to do that, rather than just counting these guys over large data sets, we need to do a little bit more sophisticated. And we notice that within each of these graphlets, there are different symmetries. So while an edge is symmetric, triangle is symmetric, square is symmetric, for example, a 3-node path is not symmetric. End nodes look the same, but different from the middle node. Also, in the 4-node path, end nodes look the same, but different from the middle nodes, and the middle nodes look the same. And these are so-called automorphism, or there's a little bit of algebra and group theory there, but let's not talk about that. Let's forget all about that. And let me just give you an example how this actually works. So let's say if you have a node, rather than just counting how many other nodes it touches, or how many other proteins it interacts with, so which we do count, so there's 4, we count other structures. So how many of the 3-node paths it would touch at an end, how many 3-node paths it would touch at the middle, then how many triangles it would touch, and so forth. Okay, so for all of these substructures. So rather than just having a one number describing the neighborhood around a particular protein and protein interaction network, we have many numbers. Okay? And we're trying to figure out, so basically, remember, we're trying to learn something biologically and medically relevant from the wiring patterns of these data. And there is a lot to do there. Okay, so let me just finish this animation. So we have a vector of numbers describing very precisely the topology, local topology around these nodes. So around this node A, we have this set of numbers, this vector, and we do that for all other nodes in the network. And we have this matrix. Okay. Now, we will go along the columns. And the first column will be your standard degree distribution. So basically, here are the degrees of the nodes in the network. Basically, you know, and this is the number of nodes having that degree. So here we said we have one node of degree 1. We have 4 nodes of degree 2, and so forth. But rather than just having degree distribution, now we have graphlet degree 1 distribution, graphlet degree 2 distribution, and so forth. Now, instead of just one distribution, we have many distributions describing the structure of this network. And now, when we are presented with another network, we find their distributions, we compare those distributions, and we have a similarity between two networks. And this is a very large number. This is very many numbers describing networks. So because, as I told you, everything is approximate, this is our approximations, but we try to constrain the data, the networks, in as many ways as possible to be more sure that they're actually structurally similar. Okay? So that is the statistics about the networks. Based on graphlets. Graphlets have been used in many domains. Now, how do they fit together? I still haven't told you that. So basically, there are redundancies, but also dependencies between graphlets. And many people get scared or think this is a big criticism of the technique, that there are these dependencies. Redundancies we'll get rid of, but they're dependencies. But actually, this is good. And this is exactly what we exploit, because these dependencies are data set driven. Okay, so different types of data will have different dependencies, and this is how we'll discriminate them based on that. So how do these guys fit together? So let's get rid of the redundancies first. So I'll show you just a simple way how we do that. So let's say C0 is the degree of a node, basically how many other nodes, what's the degree of a node, how many other nodes it's linked to. So around the node A, basically we have C0 other nodes. And then the neighbors, so basically C0 other neighbors, C0 neighbors. Now, the neighbors of a node can either be connected or not. Okay? Now, if by C2 we measure the degree of orbit 2, which is here, so basically how many 3-node paths a node touches at the middle, and we see 3, the degree of orbit 3, so basically how many triangles a node touches, then the number of possible ways the two neighbors of A can interact is C2 plus C3. So this is very simple algebra. Okay? And then once we basically enumerate all of these, we can get rid of 17 out of 72 variables, so redundancies are gone. Redundancies just blur our picture. It's not good to have them. But now the dependencies are still there. So this orbit 21, in it contains orbit 0, orbit 5 this way, orbit 2 these ways, orbit 1 that way, orbit 7 by this claw over here. So now how do we exploit this? Again, for each node of the network, we have the same vector that I just told you about. We go along the columns, and we just do Spearman's correlation between them. And in that way, we have 11 by 11 matrix that is a compressed representation of the structure of any network of any size. Okay? And now, given two networks, we have these 11 by 11 dependency matrices. We find, or correlation matrices, we find their difference, and then we have the distance between two networks. And we can see here an example on random graph models. So basically, these are synthetic networks coming from canonical models such as Erdős-Ennyi random graphs, scale-free, Barabashi-Albert, and so forth. And you can see how they're clustered together. This is just MDS, multidimensional scaling-based embedding of our networks based on the distance that I just described to you. And these are some of the real-world networks. And you can see that this method groups together all of the metabolic networks. This is all metabolic networks from Keck database over all species. Also, all protein structures are together but far away from metabolic networks. These are all protein structures that we had at the time in PDB. These are 100 Facebook networks of 100 American universities. These are some computer networks, autonomous systems networks, internet. These are World Trade Network over 4 or 5 decades, from 1962 until now. So you can see that the method works very well. These are just illustrations, of course. The full validation and everything is in this paper from 3 years ago. And now, just kind of an example. We've been in almost a permanent economic crisis for a long time now, perhaps a decade. And let me just show you what we have learned from this analysis of the World Trade Network. In the World Trade Network, the nodes are countries, and the edges are trade relationships between the countries. And the thicker the link, the more volume of trade. The bigger the country, that's the bigger GDP, and so forth. Okay, so what we can do is, from the early 60s till now, we can track how the structure of these networks changed, and we can correlate that with some economic indicators. For example, here we correlated with the change in the crude oil price over years, and we see a statistically significant correlation here. So basically this means that when crude oil price changes, it rewires the World Trade Network 2 years later, 1 and especially 2 years later. Let's leave the details of that for now, but let me just show you why this method works. Okay, and I will illustrate that on some networks that we know from theory. These are so-called scale-free networks from Barabasi-Albert. This is rich-get-richer phenomenon. So basically meaning that when a new node arrives into the system, for example, you build your own web page, you will preferentially attach it to already well connected nodes, such as your university or Google or something like that. Okay, and for these networks, we know that most of the nodes are of degree 1. They're peripheral, like these over there. There is a small number of hubs, such as these nodes here, okay? And there is a little bit of clustering. There's some clustering around hubs. And let's see here. Basically, in this network, so from this graphic correlation matrix, we see 3 clusters here. So we have orbits 0, 2, 5, 7, and 8 correlated highly, 0, 2, 5, 7, and 8. And you can see that these would be the ones that are characteristic to hubs. Okay, basically intermediary nodes connecting the nodes that don't interact. Then we have another cluster of orbits 1, 4, 6, and 9. 1, 4, 6, and 9, and these are our peripherals. And we know that most of the nodes are peripherals in these networks. And there is a small cluster of orbits 10 and 11, 10 and 11. This is the clustering around the hub. Okay? So basically, you can just inspect this and find out about the structure of your network. And we see that another type of network, such as random geometric graph, is very different. Okay? Now, for World Trade Networks, this is just an example from year 2010, but all of them look the same. We have 2 big clusters. So we have these orbits correlated by no correlation between those orbits. So let's see what these are. Orbits 0, 2, 5, 7, 8, 10, and 11. And you see these are basically the mediators in trade between non-trading countries. We call them brokers. And we see another cluster of nodes 1, 4, 6, and 9. 1, 4, 6, and 9. You see these are peripheral. So what this means is that a node is either a broker or a peripheral, but not both, because you don't have correlations there. And now you can take a network from another domain, such as the metabolic network of human, and you see that it looks completely different. With this heuristic, you cannot understand it at all, and you'd have to build another heuristic, which we did, and we published that end of last year. Now what you can begin to do, for example, you can do canonical correlation analysis, where you have a linear combination of these graphlet orbit degrees and the linear combination of economic indicators, such as balanced currency account, GDP, level of employment, and things like that. These are from UN ComTrade database, and then you are maximizing the correlation. And this is an illustration of the result. You see that these guys who are brokers in trade, so a country, again, is either a broker or peripheral, but not both. The brokers of trade are very correlated with indicators of economic prosperity, such as large GDP, large population, large level of employment, and so forth, while being a peripheral is not good. Basically, if you are peripheral, you tend to be in debt, and your economy is not doing very well. And now to track the dynamics of the countries, what's happening over the years, we can get the coefficients from canonical correlation analysis of these brokerage orbits and peripheral orbits as well. And this is an illustration of the results, again, from the 60s till now. This is just an example of 5 of the world's largest economies, and this is their brokerage score in trade. And we see that early on, basically the US has been the largest broker in trade until very recently. Early on, Great Britain was on par with the USA. This is the time when Great Britain loses colonies, and we see how its brokerage in trade falls. This is the time when it enters the EU, and it recovers and gets back together with the brokerage scores in France and Germany, and this is where basically it starts trailing off and it doesn't recover. We see that France, what happened with France and Germany, they were pretty much on par for a very long time. This is unification of Germany. And that's basically when Germany takes over as the largest broker in trade in Europe. Another interesting phenomenon is China. This is when China starts adopting the laws that enables it to enter the World Trade Organization. This is its entry into the World Trade Organization, and we see its brokerage in trade skyrocketing and passing that of the United States in 2009, actually. Okay. Now, you can maybe think, maybe this is just due to the volume of trade, but it's not. This is the volume of trade over the years, over those countries, and you can just see that we are producing more, trading more, consuming more, but you cannot see these historical effects off of these. You can only see perhaps a little bit of the recent economic crisis of 2009. And the same holds for biological networks. Basically, the positioning of a protein in a protein interaction network is indicative of a particular biological function. Here we tested over 2 species, yeast and human, because they are the most highly explored. We have the largest interactomes for yeast as a model organism that is the easiest to manipulate, to learn about human biology. But because these examples are much harder to describe, I decided to give an economics example this time. But we can see, for example, the membrane proteins, they're basically receptors of the signals. They're sensing stuff outside of the cell, and then they're starting this cascade of the signal transduction pathway to cause changes in the nucleus, to turn on or off particular genes by which the cell reacts to the external environment. So these membrane proteins, they are characteristic of these orbits that are hubby-like, while transcription factors that actually regulate how other genes are turned on or off, they're more hubby-like, they're more dense, they're more cliquish. And over the years, we have learned many interesting things. For example, this is in a protein interaction network, that similarity in the wiring patterns around proteins means similarity in biological function, membership in the same protein complexes, the same subcellular localization, meaning whether it's in the nucleus or whether it's in the cytoplasm and so forth, the same tissue co-expression in human and involvement in disease. And you can see that it does not have to involve with the neighboring ones. A protein can be far away in the network from other proteins, but as long as there is similarity in the wiring patterns, it means similarity in function. Also in this way, by only analyzing human protein-protein interaction network, we found new members of melanin production pathways that are important for studying skin cancer, and our predictions were phenotypically validated by siRNA screens of our collaborator Anna Ganesan at UC Irvine. This is where I started my tenure track in 2005. Another big problem in this area is so-called network alignment. This is analogous to sequence alignment. We want to see similarities and differences across different networks. But as I already mentioned, this is computationally intractable, and this is just an illustration of the progression of different network aligners that all have to be approximate, all have to be heuristic. This is one of the earliest ISO rank that is inspired by Google's PageRank. This is from Bonnie Berger's lab at MIT. mathematics department. And this is some of our aligners. Basically, over the years, we got them better and better. And this is one of the best performing ones that we have so far that aligns almost all yeast proteins onto human proteins. We generalized everything to directed networks. And basically now, with this generalization, we can track the dynamics in different domains, and we can analyze, for example, that metabolic network of human that you saw could not be analyzed with the previous heuristic. Once you add directions, because metabolism is directed, basically it's a chain of metabolic reactions in the cell, then this works for metabolic networks as well. And about maybe 8 months ago or so, my postdoc Noel, who is here in the audience, and I have written this perspective for science, where basically commented on a paper that was published in that issue of Science by a collaborator and a good friend, Jure Leskovec from Stanford Computer Science, where he used these graphlets to make a very robust and good measure of network structure. And basically here we also point to the directions ahead that hopefully I will talk about. Also late last year we published with Charlie Bloom's lab from the University of Toronto a paper on the analysis of his largest screen, basically covering almost all pairs of the yeast proteins. He did these epistatic interactions, basically double mutants, and observed the phenotypes of yeast cells when there is a double mutation in 2 genes as opposed to 1 and 2. And we did computational analysis linking its structure to function with them in that paper. Graphlets have somewhat taken a life of their own that I wasn't aware of at all. Until about a year ago, I went to one of these statistical physics conferences. It was at Ohio State University. And then basically I found out that totally different communities use these graphlets. For example, for image categories recognition, for scene classification, even for photo cropping, for analysis of social networks, and for personality and affective states recognition. So I had to go at least to the abstract to see what's going on. And here for photo cropping, basically, they're constructing regions, a region adjacency graph of different regions of the photo that represent, and then graphlets represent global aesthetic features of each photo, and then they use them to capture local aesthetic features. So I was like, wow, this is used for something pretty, actually. Totally unexpected for me. And this is where, in a company, they're trying to recognize personality states of people in order to maximize productivity, of course. Okay, so now let's see what do we do with multiple layers of data and how what we have done so far helps us with that. Okay, I will first talk about this patient-centered data integration. So basically we are here now, and how within the same framework we can do multiple tasks in precision medicine, from stratification of patients into clinically significantly different groups, to biomarker discovery, drug purposing, and so forth. And I'll only show example in cancer, but we do this for arthritis and cardiovascular disease, and so forth. We apply to many domains. Okay, so here we have 3 types of objects. We have patients, we have genes, and we have drugs. 3 different types of objects. So we took 353 records of series of ovarian cancer patients from the Cancer Genome Atlas, so this is TCGA database. These are their somatic mutation profiles, so basically mutations in genes of these particular patients. Now, on genes, we have different types of molecular networks that we downloaded from BioGrid and KEGG. We have protein-protein interactions, genetic interactions, and metabolic interactions. Then we have drugs from Drug Bank, and we constructed a drug-chemical similarity network on them. And also, from Drug Bank, we know drug-target interactions. We know which drugs bind to which proteins in a cell. Of course, all of this data is a bit noisy and stuff like that, but whenever we're dealing with any data, we make sure that the methods we design are robust to a large percentage of noise. For example, what I showed you so far is robust. It gives consistent results, even if you have up to 70% of noise. Okay, So now, how do we do patient stratification, driver gene prediction, and drug repurposing in this particular example? And we published this in the Pacific Symposium on Biocomputing last year. So we want to co-cluster 3 different types of objects: patients, genes, and drugs. And we do that by a machine learning approach falling generally into the non-negative matrix factorization category. And I just came back from Canada, from Beers, Banff International Research Center, where we had a large conference on large-scale molecular networks, and many people use factorizations to try to cluster their data. But they do factorizations of one matrix that represents the same data type. So this is for homogeneous data. So what you do is you have your data, okay, and then you approximately represent those data as a product of 2 factors, low-dimensional. And this is basically equivalent to a relaxed form of k-means, where F contains the cluster centroid, gene contains the cluster membership indicator, so which entries of the data belong to which clusters, and K is the number of clusters, which is much smaller than the size of the data. And this is fine if you want to cluster homogeneous data. But when we have heterogeneous data, as you saw, patients, genes, and drugs, we want to do something else. We have to go into a 3 factorization. And these approaches are very, very new, only a few years old. So what we do here, so basically, so these are somatic mutations of patients, okay? So basically, these are patients to genes. You know, each dot is a mutation of this patient in these genes, okay? We decompose that into 3 factors. We want them to be non-negative to have the cluster interpretation. We want to integrate the topologies of these networks. This is why we add constraints. So this is semi-supervised machine learning method. And we want 3 factorization because we want to simultaneously co-cluster patients and genes. So in this cluster, we'll have a cluster of patients, cluster of genes, and this is a compressed representation of this data that provides the links between these clusters. So we represent that as the product of these 3. This one, drugs to targets, basically drugs to the molecules they bind to, by another one. And basically now taken together, you know, we have the composition of R12, of R13, but we are keeping this factor the same to ensure data fusion. Okay, so this is what we just saw. Now, how do we do this? And this is a classical optimization. Well, actually, this is another computational intractable NP-hard continuous optimization problem. All we're doing here is, again, approximately solving it. We basically need to minimize the distance between these under the guidance of constraints. I mean, let's not go through the formula. This is a bit boring for most people. The constraints come from molecular networks and from these networks of chemical similarities of drugs. So basically, everything is captured in here. How do we minimize that function? This is, again, a standard way, not just in this type, but in other types of machine learning methods. You start with some sort of initialization, sometimes random, and then you have some update rules, and you go until your function converges. So this is classical optimization. Okay, now, out of this formula that we just minimized, from it, we read off all of these things that we care about: patient stratification, driver gene prediction, and drug repurposing. And let's just give illustration. So basically, patient stratification comes from this factor G1. Here we see that we clustered these 350-ish patients into 3 relatively large clusters. The first number is the number of patients. The second number is the number of the patients that unfortunately died already. And this is statistically significant according to Kaplan-Meier survival curves. And so forth. Now, in terms of driver gene prediction, we go into the clusters of genes. We find those clusters that are already enriched in drivers. What are drivers? Drivers are genes that, when they're mutated, you get cancer. They're driving the progression of the disease. You can have a bunch of other mutations, nothing is going to happen. But when these key genes get mutated, you'll get the disease, or the disease will progress. Okay, so, and the reason why we find these clusters based on the wealth of other data, and we hypothesize that if the cluster is already enriched in drivers, that there'll be other drivers in there, is because these drivers would be different in different patients, but they will usually lie on the same pathways. And this has worked well. We got 40% validations of our about 800 predicted averages in the databases that we did not use for this fusion. We essentially left some data out so that we could use that for validation. Of course, if we did this really in clinic and had biological validations, which is what we are on the way of doing, then we would integrate all available data that's helpful. Now, in terms of drug repurposing, basically it comes from this matrix here. And this is just an illustration through so-called five-fold cross-validation, how the data fusion, the power of data fusion. So basically, five-fold cross-validation means you randomly remove a fifth of your data, 20% of your data. You run your method to predict those data because then you can see how many of the right answers you got, because you know the right answers. And if you only use drug-target interactions to predict, you're not very accurate. These are areas under the ROC and precision recall curve. And as you add more data, basically the quality of your prediction increases. And 37% of our predicted drug repurposing predictions we validated in other databases as well. And I will show you here why I chose this method. This is an illustration of how cancer is a very heterogeneous disease. We have patients and genes, and again, dots are genes that are mutated in these particular patients, and this is in collaboration with Professor Staving's group at Imperial College. And we can see how basically each patient would usually have a very small number of mutations, maybe 5 or so. There would be, here's an odd patient that has a large number of mutations. Also, each gene would be mutated perhaps in one patient, mostly 1, perhaps 2, except some of the usual suspects, such as TP53. Okay, now, this is an illustration of the fusion. We are basically, now, we want to predict therapies. We want to repurpose already existing drugs to different patient groups. So basically drugs that are given perhaps to diabetic patients, we would like to give to breast cancer patients if there is a benefit. And there is a big push for this now to save the costs and time of developing new drugs. So we are integrating the existing therapies, actually the actual therapies given to these patients, chemical similarity network of drugs, all sorts of molecular interaction networks. We know patient somatic mutation profiles, so basically these particular patients from Professor Staving's lab, and we know drug-target interactions. And basically we're decomposing all 3 of these matrices going across by sharing these matrix factors that are going, that are color-coded. And an interesting thing, the reason why I chose this method is, well, one of the reasons is because it can provide mechanistic explanations. Because I can tell to the doctor, you should give this, perhaps experimental drug or already approved drug, to this particular patient, because that patient has this particular mutation, and this drug binds to this particular protein. And there are very few techniques that can provide this. Okay, so now I will slowly wrap up. Basically, we've used this also to do disease reclassification. Basically, diseases have been classified about 100 years ago now based on the symptoms and the most affected organs. And this is still what medical doctors learn, how diseases are linked to each other. And not all of it is correct. And we've seen that in diabetes and cardiovascular disease. If you have diabetes, you're more prone to get certain types of cardiovascular disease, but it protects you from other types of cardiovascular disease. That was a big surprise when it was discovered a few years ago. Now, here the question is how much of the disease ontology still holds in the light of modern molecular level data that did not exist when we came up with this disease ontology. And basically, we use the same framework, but here we have 4 different data types. We are doing some bit fancier things. But an important result here is that we can recover 80% of disease ontology. Basically, 80% of it still holds, but 20% of it may need to be rewired. And if we manage to go this way, really properly, this would be very impactful, because this basically would mean that the textbooks from which medical doctors learn will need to be rewritten. Similar stuff with gene ontology. Gene ontology is partially experimental, but mostly done on sequence alignment. Now, how much of that still holds in the light of molecular level data? And I haven't mentioned that before, but here is, in the red, these are constraints that come from these graphlet patterns of nearby or faraway nodes. And this is how we integrate that work that we have done on single layer into this data fusion framework, and it helps. We get better predictions if we do that. And this is one of our latest papers that is currently in press with scientific reports. Because network alignment, as I mentioned, is a very difficult problem. Many methods exist. They're all heuristic, they're all approximate, and there is no gold standard, such as we have gold standard for sequence alignment, BLAST, for example. And then we investigated which aligner would be good for which data, which scoring scheme should be used to evaluate the alignments, what's the coverage of each aligner in terms of biology that they cover, basically biological regions between two networks that align, and topological regions, and what's the contribution of topology versus sequence. And we have some very interesting answers. I will not talk through the details of technicalities, but basically we can pick one biological type of validation that subsumes others. We can pick one topological type of validations that subsumes all others. But the main result here is that different aligners align different regions of networks, biologically and topologically different. But when we unify them into, so basically there's a handful of aligners that together cover basically this entire mapping space between two networks. So we provided this new tool that can provide alignment of the entire protein interaction network of yeast with the entire protein interaction network of human. And this is all biologically and topologically consistent. An interesting issue here arose because we found out that the most topologically or network structurally coherent alignments we get when we use only topological data, only the structure of the networks. The most biologically coherent ones we get using sequence only. But remember, these are all on one type of data. Now I'm talking about just one type of network, just protein-protein interaction networks. And then we asked why. Is it because the existing gene annotation are ill-suited to evaluate these heuristics for aligning networks? Or maybe is this due to methodological limitations, basically to these heuristics used to align the same type of data, protein interaction network of human with protein interaction network of yeast, or any species you like, okay? And are there better ways to combine topology and genetic sequence information? And we found out that actually, yes, there is a better way, and this is why I switched back to the single layer analysis in the middle of this multiple type of data analysis. We published at the end of last year this aligner called Fuse. This is a multiple network alignment, which is even harder problem than aligning only two networks. So here we aligned protein interaction networks of yeast, worm, mouse, fruit fly, and human. So basically over here we have topologies of each of these protein interaction networks. So these are physical bindings that are possible between proteins in this data. We also have all sequence alignments, basically sequence alignment scores all to all across the entire, this full set here of these lines. And now, just for illustrative purposes, I highlighted these, and basically we are sharing this matrix factor to ensure the fusion. So this is again the data fusion framework. Now, this is a more complicated problem because even when we find out these links across, because we want to align proteins from here to proteins from here and so forth, even when we find these higher weight ones, this is still five-partite matching problem for computer scientists in the audience that isn't hard. Basically, matching problem, well, minimum weight, k-partite matching is tractable up to k equals 2, but from 3 on, it's intractable. So you have to design a heuristic that even once you have this, finds you the best alignment of proteins to proteins. And here I'll give you an illustration how here for the first time we beat sequence alignment. So basically, in the sense that by integrating sequence and topology, we get higher agreements with already known biology. And this is biological process and molecular function from gene ontology. So basically, the dashed line is what you can get by aligning sequences of these, of the proteins across different species. And the full line is what you can get by data fusion. Okay, and this is just basically saying that our method covers the clusters of genes that it gets cover all 5 species. This is for 5 species. You can see how much, while others would only cover 2 species. And basically that we are the most biologically consistent in terms of biological process and molecular function of gene ontology. Okay, so that would be it. Future directions, basically medical data is very large. It's very heterogeneous, highly dimensional, of growing complexity. It's also noisy, dynamic. It's a different time and space scales. These are all huge problems for computational scientists. Also, as I mentioned, each data type is limited in the information it carries. Okay, and this is because a disease is really a consequence of a single mutated gene or a single broken molecular interaction. It's product of multiple perturbations of complex interactions inside the cell and across the cells. Also, in contrast, patients with seemingly the same disease may have very different molecular mechanisms of disease and reactions to treatment. And as we've seen, an example of cancer heterogeneity. And this is why we need to couple network analytics with data integration, data fusion. We need to mine the wealth of complementary data. All of these data complement each other. And we need to do those types of analysis if you want to reveal the common molecular mechanisms between different diseases. Basically, if we want to reclassify diseases or have better classification of the diseases, if you want to personalize treatment, if you want to repurpose drugs, and so forth. So the methods need to be holistic for mining all available data. But those methods are very primitive so far. Even what I've shown you so far, yes, it's the best there is, but there is much more to do. And if we really want to advance here, we need paradigm shifts, both conceptual and methodological. In terms of conceptual, I don't believe that we can get too much more from analyzing the single data type in isolation of others. For example, aligning sequences or things like that. Instead, we are proposing the data alignment paradigm, where basically we want to align all data types within the single framework to build this data-driven model of the cell, okay, that we call the integrated cell. Basically, now when we have the integrated cell that encompasses everything, new things will emerge. And one of the things that we want to do is the alignment of these integrated cells that we call the cell alignment. And the same can be carried out to higher order structures of molecular organization, such as tissues, organs, and ultimately the entire organism. And we've actually made some progress on this. Methodologically, this is a work of my postdoc, one of my students and myself, and we've designed some new mathematical formalisms for this, and we are in the process of doing biological validations of our predictions with a collaborator here from biology, from microbiology at UCL. Okay, so we need new mathematical formalisms that are capable of capturing the intricacies of the multi-scale organization of the data, stochasticity of the data. But not only these mathematical formalisms, some of the more formalisms already exist, such as hypergraphs, multiplex networks, implicit complexes that come from algebraic topology and so forth. But we also need to design algorithms to extract information out of these formalisms. So even once we have a formalism, such as abstract simplification complex, we are still largely lacking the algorithms. And this is what we need to do. Now, computational issues are here to stay with us. I mean, these are just some better ways to do it, but unless somebody proves that P equals NP, or unless there is a major breakthrough, perhaps in quantum computing, the computational intractability is to stay with us, coming from large sizes, heterogeneity, noisiness, complexity, and different time and space scales of the data. And in the end, I'd like to thank the founders, and of course, my group members, without whom none of this would have been possible. And thank you very much for your attention, and I can take some questions now.
Do we have any questions?
The final code, future one, is coming back to the initial code. My question is how the biological, biophysical data flashing into the graph data, I mean the node and connection may be generated by the biologists or scientists. But initially, I guess each paper or each data may not have everything. So
Right.
How you can gather or get the original graph database?
Yeah. So, I mean, data exists at different levels of molecular organization. So I've shown you, for example, physical bindings between proteins, but that's not the end of the story. Proteins form molecular machines called protein complexes that do certain things in the cell. So you might have 50, 100, maybe even more proteins that lump together and they do certain things. They, I don't know, cut DNA in particular places, or they synthesize new proteins or things like that. Right? So that's another level of molecular organization. So biologists have already identified that for us. So we have thousands of protein complexes known. So this is another level of organizations of these binary interactions. Another type is signaling pathways that I talked about. So basically how the cell reacts to the environment. It senses the signal by membrane proteins. Let's say, okay, there is sugar here. There is sugar. Okay, I better turn on the genes that will express enzymes so I metabolize this sugar. I don't waste this sugar, right? So that there's then a signal that passes through these dynamics, bindings and unbindings of proteins along with other ligands and so forth to turn on expression of particular genes to make enzymes to digest sugar. But when sugar cuts off, then you have to stop this because otherwise you don't have an efficient system. So that's another layer of molecular organizations that's different from protein complexes. This is signaling pathways, so we already have that data. But it's not only that, but we live in an amazing time where technological advances in molecular biology are just happening in front of us. So we have single cell omics, where basically rather than just bulk sequencing all of the cells in a sample, we can now sequence every individual cell. Not only sequence, but we can do epigenetics, we can do proteomics, we can do transcriptomics of each cell individually to see what's going to happen, right? So there are all of these, and you can abstract those sorts of things into, all of these links potentially into a model, right? And then see how, and cells interact between each other through different molecules and stuff like that. So this is there. This is not something, and of course in the patient records we have all sorts of other phenomic data. It's called phenome now. It's another omic data set, right?
Is there any kind of reliable data set which is available for any user or researcher?
There are many reliable, publicly available data sets. Some of them you can download freely. Some of them you'll perhaps need a couple of months, up to 6 months to get permissions to access them. But even if my American colleagues got a full UK biobank downloaded. It took them 6 months to get permission, but they have it. Yeah.
Any other questions? Okay, well, thank you very much, Natasha. I'm going to invite Harry Hemingway to come up and give a vote of thanks.
So, Steve, thank you very much for giving me the opportunity to give the vote of thanks, and I really sincerely find this a privilege and an honour to do that. I think it's fair to say that I'm here precisely because I do not share in deep understanding of every slide that's been put up here. I do not. I'm here because I'm interested in big data and health. And the wonderful convenience that these humans, the new model organism, walk into NHS institutions and get care, good care, and they get a whole range of things that are done that collects, if you like, phenotype, phenome information on them and fuels Natasha's fantastic science. So I head up the Fire Institute in London and Institute of Health Informatics here. But before I embarrass Natasha with some wonderful compliments, I'd like to ask her which particular node on that graph she is and whether she's a big central USA node of a growing scientific network. I suspect she is. I just wanted to thank the leadership in computer science for bringing her here. And that's John Shaw Taylor, Anne Blanford, and many others that really have stepped up and brought outstanding talent to this department. I'm minded actually of Derek Roberts. Some of you may know this story. The building here we're in is named after Derek Roberts, a former Prof of UCL, who was characterised by, when bringing talent to UCL, if there was a tie break around the table, say, look, these two professorial candidates are really great, Derek would just say, well, I have both of them. But I don't think there was a tie break for Natasha's appointment. So I want to make just 3 brief points. The first point is, Natasha, we need you. We need you. And one example of why we need you is because of Eroom's law. Eroom is a palindrome of Moore's law, the law that you all know. So Eroom's law relates to the staggering, declining return on investment for drug development. So per $1 billion US, the number of drugs brought to market is now less and less each year, and now is less than 1. And we need new drugs. There have been 1,400 drug development programs in dementia, 1,400 failed drug development programs in dementia. There are about 14,000 diseases that we identify in International Classification of Diseases, and only about 600 drugs that we need. So we need new approaches, and we've heard about some from Natasha. My second point is we need you now. You have an impeccable sense of historic timing. So Barack Obama announced the new era of data-based and more precise medicine in 2015, presaging your inaugural lecture and your professorial title. In a couple of weeks' time, this country is going to publish its new UK Life Sciences Industrial Strategy, which will have data and many of the aspects here centre stage and probably with some money attached to that. It doesn't quite say your name on the page, but you could substitute your name on the page of that industrial strategy. And the third aspect of the impeccable timing is that this country is establishing Health Data Research UK, which is a core-funded national institute this year. It has funding from EPSRC, Medical Research Council, Wellcome Trust, and 7 other funders. And there will be a national competition to get at that funding and to be partner centres for that national institute, which will be a new organisation. I think Natasha's work clearly is going to have leadership roles in that national institute. So the third and final point I would like to make is that we, and this is a wide we, and I've partly got my School of Life and Medical Sciences hat on here, need to fuel your science. I think in at least 2 ways. Firstly, in bringing more data to you, particularly in the health record that I've mentioned. And secondly is people. If I had been, I was an 18-year-old medical student, if I had heard this talk as an 18-year-old medical student, I'd have had a very different career. So I'm going to say something slightly cheeky and slightly rude about my medical colleagues, so don't tweet it. But I'll start off with some facts and some quotes. So Sir George Godber, who was a former chief medical officer, famously said, doctors can count up to 1. And what he meant by that was, it's true, that they could say yes, no, disease, diagnosed or not. Hypertension, high blood pressure, yes or not. They couldn't actually get as far as to think about the value of the blood pressure. So that's 1. And then the evidence-based medicine movement in the last 2 decades essentially invites doctors to count up to 2. So you've got 2-by-2 tables, treatment, outcome, yes or no. I think what we've heard here is that doctors, amongst a wide range of scientists, should be able to count up to at least 11 in those 4-bit dependency tables. So I'm going to wrap up there and hand over to far more important speakers, Dr. Terry Hinton, who's the trustee of the Prostate Project, and Dr. Mike Hall, who's from the Prostate Computer Project, both of whom are cancer survivor patients. But just before I do that, I would like Natasha to come back in 3 years' time and represent those World Trade Network graphs. So, please, Terry Hinton.
here and listen to this exciting work that we've been stimulated about, particularly through Mike's interest. But just a few words about the prostate project, who's providing some support for one of Natasha's projects. Prostate Project Charity, a small charity started about 19 years ago, mainly retired men who are mainly survivors have got together and really raised about £7 million over the past 19 years. Relatively small. Our main objective was to improve diagnosis of cancer. As with all cancers, early diagnosis is critical. And we still have 30,000 men being diagnosed with prostate cancer a year, and 10,000 of them, 10,000 prostate cancer patients dying each year of that problem. We've been involved in supporting research at the University of Surrey. The University of Surrey at one stage had a new building, wasn't quite sure what to do with it, and we were able to say to them that actually they ought to set up a urological oncology research group, which is now 25 people strong and doing some very interesting work. Things like developing new biomarkers for early detection, virus treatment that has the great benefit of stimulating the immune system, and development of vaccines based upon patients' own white cells. So there's some interesting research going on, and very much linked with the Royal Surrey County Hospital, where we also supported the cancer centre there. Our current activity, our main activity at the moment, is to raise £2 million for a urological oncology centre in the hospital, so we can bring all the staff together. As anybody been involved in the actual front line of health knows, there's often a lot of patients coming, there's lots of staff, but there's often not much space. And so we've been able to put up half the cost of this new urology centre, which hopefully will go live in the end of 2018. So we were very impressed with the work that you've been doing, Natasha, that Mike brought to our attention. So we helped to. We're so impressed. We are aware of through the research we support, even though we learn about it as amateurs. The amount of research that's going on in medicine, very exciting, and clearly it's happening all over the world in many centres. And to be able to bring it and do that, an analytical work that you talked to us about, which hopefully will highlight some areas of really exciting developments. We thought it was something we'd like to support, even though up to now we've only really supported things locally in the Guildford area. But all the very best, and thank you very much for taking on a challenge. I hope we meet again with lots of exciting news. Thank you.
Thank you.
Okay, so I'm going to give Natasha a letter in a minute, which is to support one postdoc for 1 year to start on the prostate cancer project. But before I do that, just let me give you a few coincidences that you may find amusing. This all started when I went to a meeting of the British Computer Society at the Royal Society, and it was for the Needham lecture that Natasa gave in 2014. And just before the lecture, I met a friend of mine who was a previous president of the British Computer Society, and I confessed to him I'd just been diagnosed with prostate cancer. And he said, oh, well, so have I been. I've been in this state for some time. So that was the first coincidence. Then we heard Natasa's lecture, and we thought, well, this sounds as if it's interesting. It might be of some help to us even, because prostate cancer patients tend to live quite a long time before things get bad. So we approached Natasa and agreed on a project, and I agreed to do some fundraising for that. Then, later on, I was sent back to UCH for brachytherapy, which is another coincidence. And then, as you know, Natasa moved to UCL about 12 months ago, which is another coincidence. And here's another one. About 50 years ago, in 1968, and inspired by the thalidomide disaster, the first biomedical PhD in engineering, which is now part of this faculty, was the control of power of artificial limbs. It's a not very interesting thesis, read it if you want to, but it's half full of computer programs written in XJOF, which were run on the London Atlas, and it was almost big data analysis of electric signals then. Plenty of echoes of what Natasa was planning to do, and that was my PhD. And finally, today is my 77th birthday, which is another coincidence. So I'm really delighted that the present today, perhaps the best I could have, to be able to start Natasa's research with this one-year promise. So here's a piece of paper, Natasa, if you'd like to come up. I'd be delighted to give you this piece of paper, which promises 50k to cover one postdoc for 12 months, and we hope we'll be able to raise some more, but we're not promising. Thank you very much.
All right. I'd like to thank the Prostate Project very much. It was really wonderful to have met you guys about 3 years ago now, and I really enjoyed our work on this, and I'm very happy about this, and I plan to start this from New Year's. We already have some ideas. We've already done some preliminary work. We're actually even beginning some biological validations of new genes that we strongly believe are involved in prostate cancer, and this will really greatly help to further that work. So thank you very much, guys. And thank you all. Thank you all very much.
Okay, I'd like to thank all our speakers, actually, particularly Natasha, obviously, but also Harry Hemingway for offering the vote of thanks, and to our generous donors as well. It's fantastic. So thank you all very much indeed.